Let's take a look at what you're doing in lab this week. This is week 4, and we're looking at ecological interactions. You have your overview. So our objectives for the week, we're going to look at a simulation of an intertidal community and analyze some of the impacts of those interactions between the species. Part of that study will be to perform a removal experiment in this simulation. You're going to go out into the field and use quadrat sampling to look at abundances of lichen. So you have your weekly lecture, and then you have your 2 assignments here. The assignment 1, the barnacle competition simulation, and assignment 2 is the lichen survey. So let's take a look at this first one. This is the barnacle competition simulation. So you have some stuff you can read through here. Basically, it's saying that if you have 2 species that are competing for the same limited resource, there are several outcomes that we can see through natural selection and evolution. One might be that one species would exclude the other. It would outcompete it, so it would drive that species out and it would take over and you'd only have one species left. That's called competitive exclusion. Another thing that can happen is you can get coexistence of the 2 species. And normally when you get coexistence of 2 species that are using very similar resources, it's because they've had a shift in their niches and they're using slightly different resources. We refer to this as resource partitioning. So if they're foraging in slightly different areas of a tree and they're no longer competing directly in the same area, that would be that type of resource partitioning. So while they might be able to use the entire area, they might be able to use from the ground up to the top of the canopy of the tree, that would be their fundamental niche. They're not using it because of competition with this other species. So let's say now each species is only using half of that area, so their realized niche, it's smaller than their fundamental niche. Their fundamental niche is what they could be doing, their realized niche is what they're actually doing. And it's, again, usually smaller because of the effects of competition. The first thing this assignment is asking you to do is a pre-lab reflection. So it wants you to think about an intertidal zone. If you've been to one before, you can think about what you've seen there and answer these questions. If you haven't seen one before, there's a video here that shows you an intertidal zone and shows you some of the organisms that live there. So after your pre-lab reflection, you're going to go to this virtual biology lab and you're going to do an experiment on barnacles. The instructions here are pretty straightforward, but I do want to show you a couple of things. In one part of the assignment, it's going to have you design an experiment to look at competition between these 2 species. And it tells you this, but I want to make sure that it's clear. When you want to remove one of these, so let's say we're going to do a removal of Balanus. If I remove them, the other thing I have to do is I have to come up here and I have to put the larva down to 0. Because if I don't, they're just going to creep back in there. So if I truly want to remove all of them, I have to remove them, but I also have to put that down to 0. So when we run the simulation, we can see the tide going up and down, and we can see the different barnacles establishing themselves. So we can see mostly the orange barnacles down here, and we see their frequency distribution over here. We see a lot of the whites up here, and the frequency distribution there. And what I wanted to point out, because I initially thought this was a bit confusing, is if we look at where our white barnacles are, they're up here. And then if we look at the mean elevation on the rock, it's 348. If I look at the orange ones who are below them on the rock, their mean elevation is 470. So I thought that was kind of confusing because to me elevation would represent height here. But it's actually calculating it from the high tide line. So it's using the high tide line and then going down this way to measure that elevation. So first you're going to run the simulation, see how it works, and then you're going to, in step 7 here, you're going to design an experiment. Change your settings in the simulation to set up an experiment where you sequentially remove each of the species that are there and then see what happens. So it's going to ask you some questions about how did you set up the experiment and then what were your observations. My recommendation is to use the snipping tool to get that information after you run it. The snipping tool you can find in your Windows menu. You can usually just start typing in snip and you'll see it pop up. It looks like this. And then you just hit new snip, just copy it. I can then copy this and paste it right into my Word file. So then you'll just answer some of the questions here about the experiment that you have conducted. And you see your rubric underneath to make sure that you've done what it's asking for. Your next assignment this week is going to be to go out and find lichen and to document what you're finding. What you're going to need is a sheet of transparency paper, so clear paper, so usually like an acrylic sheet, a Sharpie. You're going to need a measuring tape, a random number generator, which you can usually find an app for your phone for that, and a compass, or you can use the compass on your phone. You're going to select 2 different locations, and you're going to want locations that are going to have lichen on them. So lichen tend to grow either on buildings or rocks or trees, so you're going to need to find a place that has lichen. So you may have to scout it out a little bit first to find a good location. You're going to find 2 of those locations. You're going to download this grid, which looks like this. You're going to put this grid on that clear paper. So whether you photocopy it on there or you sketch it on there, but you want this grid on that clear acetate. You're going to go into an area. You're going to use the random number generator to give you a number, and that's going to tell you how far to walk. So you're going to be walking along a path. Random number generator tells you 20. You're going to take 20 steps. You're going to stop. And then as soon as you stop, you're going to find the nearest spot that meets whatever criteria. So you might always be looking on the north side of a tree or the north side of a rock. So you're going to make sure you always do it the same way in this one location. And then you're going to hold the transparency at a particular height. So if you're looking at, say, the bark of a tree, maybe it's 3 feet off the ground on the north side of the tree. So always the same way. You're going to hold that acetate up to the tree at that point. And then you're going to mark in each one of those boxes, you're going to count how many of the circles contain each of these kinds of lichen. There's a link that shows you different pictures. So the crustose is going to lay very flat against the surface. And then you've got fruticose, which will have sort of like little thin stalks, hair-like stalks that come off of it. And then you've got foliose, which looks a little bit like a leaf, a little bit flatter than the fruticose, but not quite as flat as the crustose. So you're going to be looking for those 3 types. So you're going to have a data sheet. Our very first sample, we lay down the acetate. In every box, there's 10 little circles. In that first box, how many of those little circles are touching this type of lichen? How many are touching the fruticose? How many are touching foliose? How many are touching moss? Moss is actually a vascular plant. Lichen is a fungus and an algae. And then how much of it is actually bare surface? So all of this should end up adding up to 10 because we have these 10 different dots on each one. So you'll do that for all 9 boxes on your one sheet of paper for one site. Then you're going to do that 10 more times in that one site. So you'll have 10 of these sheets for one site. Then you're going to go to your second site, do exactly the same thing. So you'll have 10 sheets for that site. So when you use this table, you're going to summarize what you just did in the previous one. So for each sample, you've got this one sheet of paper that's got 9 blocks on it, and then each of those blocks has 10 little circles. So you have a total of 90 circles. And so you're going to essentially just say in this one sheet, how many of those circles total were crustose, fruticose, foliose, moss, or bare surface. So then you can say for each sheet of paper that you looked at, what was the percent coverage by essentially looking at the fraction and then multiplying that by 100. For each site, you'll have 10 of those. So you can take the average percent coverage for your 10 samples from site A. You can take the average for site B. And then you can compare site A and site B and look to see if there's any differences and then hypothesize why you see those differences. So you're going to compare the two, create a graph for your averages. So that will typically be a bar graph. What is my percent coverage for site A? What is my percent coverage for site B on average? So you'll have a graph that looks at those. And then you'll explain the meaning of your graph. You'll try to interpret why you're seeing the differences you are. Now, it also mentions up here that you will want to take photos of each of your sites and each of your samples. In addition to your data tables and your graph, you want to include photographs of your location. Down below you have your grading rubric. So that's everything for this week. If you have any questions, please let me know.